You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that had, has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoy the podcast, please listen, subscribe, like, and share it with friends. Thank you. Hello, this is Richard Jacobs with the Future Tech and Future Tech Health podcast. I have Joy Wolfram, PhD. She's in the Department of Biochemistry and Molecular Biology at Mayo. We're going to be talking about nanoparticles, nanomedicine, and extracellular vesicles, also called exosomes. So, Joy, thanks for coming. How you doing?
I'm doing good. Thank you so much for having me, Richard.
Yeah, tell me what's the focus of your research and your work?
So I'm the director of the Nanomedicine and Extracellular Vesicles laboratory in Florida at Mayo Clinic. And what we focus on is developing nanomedicines to cure or hopefully one day cure diseases such as inflammation and cancer.
What's important about it being nanomedicine? Why? What's wrong with micromedicine? What's wrong with picomedicine? Like, why that order of magnitude?
That's a great question. Well, actually, most conventional medicines are just one molecule. So they're a lot smaller than what we think of nanoparticles. And so one of the advantages of actually having a medicine that is on the nanoscale is that we have more space. And because we have more space, we can add all these extra features to help us deliver the medication and also add extra medication into the nanoparticle. And these nanoparticles are basically like tiny cars, and the passengers of these cars can be, cancer drugs or anti-inflammatory compounds. And usually we inject them then into the blood. So the blood is like the highway where the cars are driving, and then they drop off the medication or the cargo at the site of diseased tissue.
That's pretty cool. So small molecule drugs, the nanoparticles you're working on are what, like 1,000 times bigger than a typical small molecule drug, just to generalize?
Yes, exactly. I would say 100 times bigger, because the small molecules are usually 1 nanometer and our nanoparticles are usually around 100 nanometers.
Okay, got it. So you can add functional groups, you could add, I guess, cavities to shield payloads that can be delivered. I guess you can do all kinds of stuff, right, with nanoparticles because they're bigger.
Exactly. Those are great examples of some of the things we and other groups are working on, yes.
So what's your focus within, are you good at creating the nanoparticles, but you're arming them with functional areas? Like, what's your focus in this area?
So, yes, great question. So my past focus used to be synthetic nanoparticles. So we will synthesize these nanoparticles from lipids like fats or porous silicon. But today my lab is primarily focused on extracellular vesicles or exosomes, like you heard earlier. And these are actually biological nanoparticles. So they're found in our bodies and in all biological liquids, such as urine and saliva. And so what we do is we use these extracellular vesicles for therapeutic and diagnostic purposes. So, for instance, can we diagnose cancer by looking at the extracellular vesicles in the blood? Or can we take extracellular vesicles from adipose tissue, for instance, from fat tissue, and use them for therapeutic purposes? Because we know that fat tissue has stem cells, and stem cells have anti-inflammatory and also properties that may potentially heal tissue.
So what kind of cells do you study to see what kind of exosomes they put out, and what have you learned in general about? Is it a hard task to generate a nanoparticle from scratch? Is it a lot easier to make a biocompatible nanoparticle by looking at what cells put out already and just altering them?
Yeah, fantastic question. So, well, both have their disadvantages and advantages. When you create something from scratch, of course, you have a lot more control over it. But then again, it's difficult to make so much complexity that we see in biological systems. So the advantage of these extracellular vesicles is that they're very complex and we don't have to design them from scratch. But it can actually be difficult to isolate them from cells or tissue. And we are mostly focusing on tissue. So that would be like different cells in adipose tissue or plasma, so blood. So that would be basically extracellular vesicles from every cell in the body, because all our cells are releasing these extracellular vesicles that are kind of like text messages. And so all of them end up in the blood. And when you're analyzing the blood, it's actually a mixture of cell sources that have released these extracellular vesicles. And as you, Richard, mentioned earlier, we can go back and modify them. So we can add therapeutic agents inside, or perhaps modify the surface to make them even better. But essentially, we are taking advantage of nature's own strategies, our own biological strategies that have been developed through millions of years of evolution to effectively transport molecules in the body. And so we're excited about that.
What's your sense of how many different types of EVs a given cell will put out, and under what conditions? I mean, are there like thousands of different kinds, and how do you even characterize such a thing?
Exactly. So that's something that is debated currently in the scientific community, is that how many different types of EVs are there just from one single cell type, what type of EVs are released and how do they differ. And so there's a lot that we don't know. And there are many groups around the world that are trying to figure out exactly what types of EVs there are. So extracellular vesicles, EVs, and also how we can actually distinguish one subtype from another one. So currently there's no sort of exact criteria. So different groups are using different markers and so on. But it's a relatively new field, so that's why it's exciting. But we still need a lot of knowledge in regards to those types of questions.
Do you have an idea of where EVs are produced within the cell, and then if EVs enter a cell from an outside source, where they go to in the cell and what happens to them?
Yeah, great question. So there's sort of three main categories of EVs, and one is apoptotic bodies. So when a cell dies, there's these large extracellular vesicles. Then there's also exosomes, and exosomes actually come from the cell, from inside the cell, while microvesicles bud from actually the cell membrane. So these are the main three types, but there may be additional types that are formed in different ways that we don't know about yet. And then when they're taken up by cells, they could go to different locations, but usually they could go to the lysosome, which is sort of the trash can of the cell, or they could have some type of signals that direct them to specific intracellular locations. But again, this is something that we're trying to figure out, is how does the extracellular but also the intracellular trafficking of these EVs take place. And it's been a little bit difficult because it's not that easy to label the extracellular vesicles. If you use sort of lipophilic markers, they may escape from the surface of these extracellular vesicles. So at that point, you don't really know what you're tracking. So the field, we're also trying to develop these methods to answer these types of questions.
So do we know where? So we know various places that EVs can be created within the cell, but do we know what structures create them? Are there certain organelles? I know you said there's budding of the interior membrane, I guess, but are there certain organelles that are responsible for creating them and packaging them and sending them out?
Absolutely. So that's mostly for the exosomes. There's an entire machinery. So it can go, through the Golgi or endoplasmatic reticulum, because you need different components. So you need the internal cargo, but you also need the lipids. So there's several, internal components that come together, but then you have these similar to the vesicle trafficking. When something is taken up, you also have a vesicle trafficking system when something is secreted out from the cell. And also we're still trying to figure out exactly what are the essential pathways necessary for these exosomes and also the microvesicles. But microvesicles, you can even, with physical forces or other ways, you can make the cell, form this budding off from the cell surface. So that may be less complex than this exosome sorting machinery.
And when a nanoparticle or an exosome enters into the target cell, where does it go?
So it's similar to the synthetic nanoparticles. So it can go to many different places, but one of the main places where things go after they've been endocytosed is to the lysosome, which is the trash can. But the lysosome has a lot of enzymes that destroy biomolecules. So they're pH sensitive because it's very acidic pH. So if it does end up in the lysosome, they're usually destroyed, or they escape before the lysosome. But then they can go, some of the cargo can even make it through additional barriers, such as the nuclear envelope of the cell. So it depends, and some of it also depends on the resorting of the cargo. So some groups have shown that, RNA can enter from an extracellular vesicle into a cell and then get repackaged again into a new extracellular vesicle that then gets released. So sometimes the cargo doesn't even stay permanently in the cell that it's taken up by.
What is your thought? When a particle enters the cell, what is determining where it should go, how it should be treated? What do you think is the mechanism for that? Like, how would, if you think about there's no discernment, then you just have to evade, I guess, what the cell would normally do to the particle that enters it. But if you think there's discernment, then I guess you could work within that, to tailor the particle so that it can go to the right place or be recognized in the right way. Like, what's the overall theory that you have?
Absolutely. So it's many different factors. So something as simple as shape can affect how they are processed by the cells. It's also other physical properties such as surface charge and even the softness of the particle itself. So there's different receptors on the cell surface that can sense, if you're dealing with a very soft extracellular vesicle or hard extracellular vesicle. Of course, synthetic nanoparticles, some of them like gold, is a very hard material, so it's processed in a certain way. And those are sort of the things that we don't normally think about. We normally think about these proteins on the surface, and the proteins will bind to something. The proteins on the surface of the extracellular vesicles will bind to something on the cell surface. But it's also all of these physical properties that actually can affect how the extracellular vesicle is taken up and processed. And recently it was shown that cancer cells that are metastatic actually release extracellular vesicles that are softer. So there's all these interesting physical properties. And honestly, a lot of this, is not known even for synthetic nanoparticles. There are certain uptake pathways like clathrin-mediated endocytosis that typically, result in one outcome. And sometimes just by changing the charge of these nanoparticles, can you get it to go to the caveoli pathway, which is another endocytosis pathway. But still, we don't understand exactly what is affecting how things are being sorted inside the cell.
Well, what's amazing to me is that cells, for instance, in our body are in this milieu of tons of stuff floating around. How do they know, okay, I've got I'm a liver cell. I need to talk to the heart cells, or I need to talk to this other type of cell. So I need to prepare an EV that the other cell will take in and recognize and, perform a certain action. Or if I'm taking in hundreds or thousands or, God knows how many different EVs internally, how do I know what to do with them? It just doesn't seem possible that it's just, it's random. There's got to be some, discernment mechanism somehow, or multiple.
Absolutely. Yes, that's absolutely a very good point. And the other thing is that originally, let's say 10 years ago, the scientific community really thought that the extracellular vesicles were just waste products, so that they didn't have really a functional role in communication between cells. And today we have several examples that they do. However, we don't know to what extent. So we don't know if 50% of the extracellular vesicles are just waste products and 50% are involved in cell communication, or whether it's just a small portion, such as 1% of the extracellular vesicles that are in our body that actually communicate between cells, and the rest are more of a waste system to get rid of biomolecules such as RNA. So that's a great question, and these are things that we still need to figure out.
So it's a very complicated problem. What's your targeting? Are you looking at trying to affect certain conditions? Are you looking at certain mechanisms of doing this? Like, what's your focus?
So our focus is actually twofold. So one is on the diagnostic side, and that relates to cancer and specifically to metastasis. So can we, by looking at the sugars, so a lot of people don't talk about the glycans, the sugars, and they've been sort of overlooked, but we believe that they're very important in cancer when they're on the surface of extracellular vesicles. So by looking at extracellular vesicles in patients' blood, and specifically the sugars on the surface, can we predict which patients are likely to develop cancer metastasis? And by doing that, potentially we could then, if it works, have these patients that are at risk get more aggressive treatment than the patients that are not at risk for the cancer to spread. And sort of the second direction of my lab is therapeutics. And like I was mentioning earlier, from adipose tissue. And the specific conditions we're looking at is, can we take a patient's fat, isolate the extracellular vesicles, and then use them for conditions such as myocarditis, which is heart inflammation, or lung ischemia, reperfusion injury. So when the lung doesn't get oxygen for some time, and then, for instance, the blood vessel is blocked and then it's reopened, you have a lot of inflammation. So can we use those extracellular vesicles from the fat to lower this? And within this context, can we also load them with the conventional anti-inflammatory therapeutic agents? And so we get combination therapy. So we get the endogenous anti-inflammatory properties from the vesicles, in addition to then getting the effects from the small molecule that we have loaded inside the adipose extracellular vesicles.
Well, in terms of communication, have you looked at communication going on between, I'm talking about cancer. So the primary tumor and metastases, have you looked at what kind of communication is going on? Do you believe there is communication going on between the primary tumor and the metastases, the metastatic sites?
Exactly. So we are currently looking a little bit into that, but there are other groups that have definitely already in 2015 shown that the primary tumor can release extracellular vesicles that then go to distant organs, and they prime the organ for the cancer cells later to metastasize from the primary tumor, so they contribute to this pre-metastatic niche. And so I do think that some of these studies are convincing, and that there could be definitely a major role that these extracellular vesicles play in the metastatic process. And so that ties a little bit into our research in cancer diagnostics is, this happens before, or we believe that it may happen before the cancer cells metastasize. So it's a good way to potentially predict which patients will develop metastasis by catching these extracellular vesicles in the blood when they are released by the primary tumor before the metastatic process.
Yeah, or even when it goes on, if you were able to cut off communication, if there is communication, I wonder what that would do. And is there a way to tag exosomes yet and see where they go in the body and what other cells they go inside of?
Exactly. So I touched a little bit upon that is with the use of these lipophilic dyes, fluorescent dyes, and that's the most common method. But again, not very reliable, because these dyes, they can detach from the extracellular vesicles after you've injected them. And then if you see a fluorescent signal, you don't really know where it's coming from. So more sort of advanced techniques are radio labeling. So you have a radioactive probe that can tell you where the vesicles are going. And some even inorganic molecules can be loaded inside the extracellular vesicles, and you can detect them in that manner. But it's challenging. It has been challenging. But a lot of groups around the world are developing improved methods to track these vesicles inside the body.
Yeah, I wonder if any cell has like a homing pigeon type vesicle where they send it out, it does something, gathers info, and it comes back to that cell. I mean, that's pretty crazy, but I don't know, just the thought occurred.
No, that's fantastic. Yes, definitely. Maybe not the exact same membrane, but the cargo may be coming back, because we know it's already going through multiple cells and it can be repackaged. But I haven't seen any reports of then it returning, that same cargo returning to the same cell. But that would be a nice feedback loop there. So potentially, if we wait a few more years, then maybe you're proven right.
So where do you think the initial breakthroughs are going to come from? What part of your study do you think may hold the key, the most promising area where you think you're going to make a breakthrough?
Well, I think, what I'm excited about is regenerative medicine applications, because in general, that's a emerging field. And there's a lot of unmet patient needs that we could potentially solve by developing regenerative medicine. And there's already, I think, 4 clinical trials around the world, not by us, that are looking at extracellular vesicles for various regenerative purposes, such as, diabetes related wounds and even diabetes itself. So how can we restore damaged tissue? So I think that's an exciting avenue to explore.
Well, I guess for a real head trip, do you think that our microbiome is sending out EVs and receiving EVs, and those are talking to our, somatic cells? Do you think there's communication there?
Oh, absolutely. I think it's been shown. It's not my field of research, but I know that there is communication between the extracellular vesicles from, let's say, the bacteria and fungus in our body with our own cells. And I think that type of communication has also, developed through many years of evolution, where we have this synergistic relationship between the microbiome and our bodies. And I'm sure some of that is through extracellular vesicle transfer.
And what about what if all these cell types, let's say in the human body, were characterized, their membranes, what they look like, what proteins were there, et cetera? Perhaps then a nanoparticle could be created, or an EV could be co-opted, and it would target only a certain kind of cell, because we'd know that's the only type of cell that has this structure on its membrane, and the EV or the nanoparticle we made will only get in through that kind of a portal, so we can target that cell specifically.
Yes, potentially. That's a great idea. I think we're still far away from that in mapping exactly all the membranes in the body and understanding how they work. And also, the extracellular vesicle membranes don't necessarily correlate with the cell membrane, especially if they're exosomes. So there could be a lot more intracellular and extracellular membranes apart from the cell membrane that could be characterized. But I definitely think, down the line that could be possible, and we would probably need a lot of computational tools as well to generate and analyze that much data. And a lot of these membranes are not static either, so they're dynamic, and they're, actually the cell membrane, it's usually you think of it as, something that just sits there, but it's actually a lot of movement, and there are a lot of biomolecules moving all the time on the surface, and just how they move, the direction, and the speed they're moving, all of that could be very important for biological interactions.
It's like mind-blowing. Like, have you ever tried to, imagine yourself as a cell, what you would see on the outside, what you have going on the inside, and all the, again, I'm anthropomorphizing it, but all the decisions and the discernment you'd have to make in interpreting all this stuff going on. There's thousands or millions of things outside of you.
Exactly. And I know of some people that have even created like dance groups that the dancers try to mimic what is going on inside the cell. So there's a lot of exciting things that we can do for science education is by imagining what is happening in the cell through using also the art.
So what's well, I guess there's just so much to ask. What do you think is possible in the next 5 or 10 years, in particular with your research? What headway do you think that you can make, or what would be a fantastic result if you can achieve it?
Exactly. Well, as you know, Richard, it takes 12 years usually. That's the average to get something from the lab into the clinic, and that's around $2.7 billion. So it is a marathon, and that's if you're successful. And over 90% of what we develop ends up failing at some stage before it reaches the patient. So I think there's a lot of sort of intermediate success stories potentially. My career goal would be to get something into the clinic to help patients, but there's never any guarantees. But in general, for the field, I think already now and in the couple of years ahead, we are seeing a lot of cosmetic applications for extracellular vesicles, and these cosmetics are not necessarily regulated at all, which makes it faster, but there can also be a lot of ethical issues when you're injecting things into the body, even though it's not for medical purposes. So this also depends on the country that you're looking at. But I already know that there are some lotions on the market that have extracellular vesicles in them. So we're already starting to see those products.
Oh, what have you seen as the effects of those lotions? Did you get any insight from seeing them and seeing what they do?
The one example I know is from a Korean company, and I believe that the science is quite solid, but the problem is that the dose of extracellular vesicles in the lotions is too low. So at the moment, the dose would probably not have any biological effect. But then if they could increase the dose, but that would also significantly increase the price of these lotions, there could potentially be some effects. And this is from mesenchymal stem cells. So these are the stem cells that are releasing the extracellular vesicles.
Yeah, for a topical application, how many EVs would you need? Like millions or billions or trillions of them? And then if you were to inject something that would go through the bloodstream, is there any sense of how many EVs would be needed to accomplish the therapy?
Yes, there is. So it varies, but usually it's depending on the species. It's usually something like for 10 to the 7 to 10 to the 9 vesicles, even 10 to the 12. But actually in the human body, the plasma itself has 10 to the 9, so billion vesicles per milliliter. So probably that's sort of what the cells are exposed to in our body. So probably you need a dose to kind of compete with what is already in the body. And as you mentioned earlier, there's so much going on. There's so many extracellular vesicles from all these cells being released. So dose is always an important question.
Yeah. And have you seen examples of someone, with cells in culture and putting them through different paces? I don't know, like changing the pH, seeing what vesicles they put out, changing the temperature, putting a food source. Has anyone done something like that to just try to characterize, stimulus response in terms of EVs that are put out?
Yes, absolutely. Again, amazing questions here. Yes, so one of the sort of main things is that when a cell is under stress, so whether that's, lack of oxygen, lack of, nutrients, or if it's mechanical stress, like you're pressing on it, any type of stress tends to usually increase the amount of extracellular vesicles. And others have also tried, giving various molecular compounds to extracellular vesicles, such as like inflammatory compounds, to see how that changes the amount and type of vesicles. But there's still so much that we don't know, and there's many interesting studies we could do, especially like with pH and so on.
Okay. Well, very good. What's the best way for people to find out more about nanoparticles and EVs in general, and then in specific? I don't know if you wish any contact to your lab or anyone to find you.
Absolutely. So you can learn more by checking out my TED Talk, and then you can contact me through email as well. So my Mayo email is available online. So I enjoy hearing from the colleagues in the scientific community, but also from the general public.
Well, that's great. Well, Joy, anything else that I should have brought up that I didn't include?
Well, in general, I think it's, unrelated from extracellular vesicles, but related to the scientific process and science is that, we need more representation from diverse scientists. So we need to support women in science. We need to support racial minorities. And that's something so important also on the intellectual level, because if we have people that think in the same way, we're not going to have that many new ideas. But if we have people from different backgrounds, different races, different genders, we're going to have a lot more creativity, and we're going to be able to solve more challenges because we're coming at it from many different viewpoints and also different leadership styles that are necessary to make progress for extracellular vesicle research, but for any field of research.
Okay, well, very good. Well, I appreciate you coming on the podcast. Thank you for being here.
Yes, thank you so much, Richard. And I'm very impressed by the scientific questions.
Oh, thank you. I'm a good questioner. I don't know if I have answers, but got a lot of questions. So.
You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoyed the podcast, please listen, subscribe, like, and share it with friends. Thank you.